the mitochondria contains its own DNA. This DNA is present as a small circular DNA molecule of 16,569 bases. The DNA contains 37 genes, 2 ribosomal RNA genes, 22 tRNA genes and 13 messenger RNAs that code for some of the proteins that are found in the mitochondrial respiratory chain complex, the MRC. The subunits of the MRC that the mitochondria cannot make are produced from the DNA in the nucleus. And this raises an interesting question of how the mitochondria communicates with the nucleus to make the subunits it needs. The human mitochondrial genome is not like the nuclear genome. Each mitochondria can contain between 2 and 10 copies of the genome, and these genomes can have different sequences. That is, there can be either homoplasmy, which means that all the mitochondria have the same DNA, but that is rare, or, which is much more common, heteroplasmy, where there is a mixture of mutant and wild-type mitochondrial DNAs. The mitochondrial DNA has no introns. Mitochondria are predominantly maternally inherited. Mitochondrial DNA is not replicated in synchrony with cell division. And there are also differences in codon usage between the nuclear genome and the mitochondrial genome. For example, UGA is stop in the nuclear genome, but except for in plants, where it still is stop, in the mitochondrial genome it is tryptophan. As mitochondria have their own DNA and make their own proteins, then mitochondria can have genetic disease. When mitochondrial disease occurs, it will be passed down the maternal line as we get pretty much all our mitochondria and hence our mitochondrial DNA from our mothers. Consequently, standard genetic crosses will not remove the mutations from the gene pool. When mitochondrial disease occurs, it usually impacts tissues that have a high energy demand, such as the nervous system, muscles, and the brain. The only current way to treat mitochondrial disease is through 3-parent babies. In 3-parent babies, a donor egg has its nucleus replaced with the nucleus from an egg from a mother that has mitochondrial disease, hence effectively replacing all the mitochondria that contain the mutation. The egg is then fertilized with the sperm from the father.